Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
In the world of terpene chemistry, abietic acid has always been a stubborn feedstock, chemically rich, but frustratingly hard to diversify with precision. What makes this paper exciting is that McCadden and colleagues show how Streptomyces can be turned into a living chiral factory, carving abietic acid into a portfolio of new, oxygenated scaffolds that traditional catalysis would struggle to reach cleanly. Scientific breakthrough. The core idea of biocatalytic diversification of abietic acid in Streptomyces is deceptively simple. Use the native oxidative machinery of Streptomyces strains to generate a suite of structurally distinct abietic acid derivatives in a single biocatalytic platform, instead of engineering bespoke chemistries for each target. The authors express or harness Streptomyces enzymes, likely P450 monooxygenases and related oxidative biocatalysts, to introduce regio and stereoselective oxidations and rearrangements on the abietane skeleton, generating new molecules that expand the chemical space of resin acids. Based on the title and placement in JIMB's biocatalysis portfolio, the novelty lies in showing that wholesale Streptomyces biocatalysts can perform multiple, complementary transformations on abietic acid, yielding diverse products rather than a single, over-oxidized endpoint. The study does not explicitly report all reaction mechanisms in the accessible summaries, but it clearly positions microbial oxidation as a route to new abietic acid-derived scaffolds with potential biological activity. From a bioprocess standpoint, this work treats Streptomyces not as an antibiotic factory, but as a heterologous oxidation platform for a hydrophobic diterpenoid substrate. The practical challenges are classic: low solubility of abietic acid, potential toxicity to the cells, and the need to balance growth with bioconversion so that oxidative capacity is high when the substrate is added. Although the detailed methods are not visible in this interface, comparable Streptomyces biocatalysis studies typically rely on wholesale resting or semi-resting cultures, phase transfer or co-solvents for substrate delivery, and careful control of aeration to support high P450 turnover, elements that are consistent with a biocatalytic diversification strategy. The key process insight is that you can run this as a biotransformation rather than a de novo fermentative pathway, feed abietic acid into a pre-grown Streptomyces culture and let the cell's oxidative machinery do the heavy lifting. The abstract-level information accessible here does not list explicit titers, space and time yields, or conversion percentages for each abietic acid derivative. What is clear is that multiple oxidation products are generated and structurally characterized, likely via NMR and MS, demonstrating that the Streptomyces system delivers more than a single major metabolite. Past work on abietic acid oxidation has reported modest chemical yields and mixtures of isomers under non-enzymatic conditions. This study highlights that biocatalysis can deliver defined, stereoselective products, even if the exact molar yields are not spelled out in the summary. The paper therefore reads as a discovery and proof-of-concept platform piece rather than a fully optimized production process. It establishes that meaningful conversion to novel derivatives is achievable, but the study does not explicitly report optimized volumetric productivity or scale beyond laboratory flasks or small bioreactors. For listeners, the important takeaway is that the system is robust enough to generate an analyzable suite of new compounds, which is the necessary first step before serious process intensification. From an industrial perspective, the real power of this work lies in its ability to open up chemical space around a cheap, bio-based feedstock, abietic acid, using enzyme selectivity rather than multi-step synthetic campaigns. Abietic acid is abundant in tall oil and other resin streams from the pulp and paper industry, making it an attractive, renewable starting material if you can reliably transform it into higher-value derivatives. Biocatalytic diversification in Streptomyces offers a route to oxygenated abietane derivatives, potential anti-inflammatory, antimicrobial, or materials-relevant compounds, with less protecting group gymnastics and milder conditions than classical organometallic or radical oxidations. At the same time, the paper is honest in its scope. It does not present full techno-economic analyses, scale-up case studies, or downstream processing flow sheets. Those remain to be built on top of this platform. Regulatory implications will depend heavily on application, pharmaceutical, cosmetic, or specialty chemical, but a microbial biocatalytic process using Streptomyces is squarely in the realm of established GMP-compatible organisms, provided host strains and impurities are controlled. The market context for abietic acid derivatives is fragmented but promising. Resin acids and their oxidized analogues appear in coatings, adhesives, inks, and as scaffolds for pharmaceuticals and agrochemicals, with global resin acids markets estimated in the low billions of dollars and specialty derivatives commanding significant price premiums. The paper itself does not give market size or CAGR numbers, but its focus on the diversification strongly aligns with the demand for libraries of natural product-like scaffolds in drug discovery and high-performance materials. External analyses of biocatalysis in fine chemicals suggest robust growth, often high single-digit to low double-digit CAGRs, driven by green chemistry mandates and the need for chiral and functional group-rich building blocks. In that context, a Streptomyces platform that can convert a low-cost, renewable diterpenoid into a set of distinct, structurally complex molecules is strategically attractive, especially if downstream biological screening identifies potent bioactivities. The study does not explicitly tie individual derivatives to specific markets, but it clearly positions abietic acid as a versatile starting point for value-added chemistry rather than a bulk resin byproduct. For founders, this paper is essentially a technology teaser. Biocatalysis can unlock a rich portfolio of abietic acid derivatives, but the value creation lies in connecting that chemistry to specific use cases and scaling the right transformations. One pathway is a resin-to-scaffold platform company that partners with pulp and paper producers, taking crude resin streams, enriching abietic acid, and running Streptomyces biotransformations to produce libraries of oxidized biotanes for pharma and agro discovery programs. Another is a more focused play: identify one or two high-value derivatives with attractive activity or materials properties, then optimize the corresponding Streptomyces biocatalyst for industrial titers and yields, wrapping process IP around strain selection, expression systems, and biotransformation conditions. IP potential is strong. Specific enzyme sequences, engineered Streptomyces strains, and process conditions that yield defined abietic acid derivatives are all patentable. Although the article's accessible material does not list patents, the very framing as biocatalytic diversification suggests an eye toward platformizable chemistry. For investors, the risk-reward profile is classic for biocatalysis. Technical risk lies in scaling wholesale biotransformations with hydrophobic substrates and demonstrating clear product market fit for at least one derivative. The upside is a renewable, defensible source of complex chiral molecules that are otherwise expensive to make. Stepping back, biocatalytic diversification of abietic acid in Streptomyces is less about a single molecule and more about a mindset: treat abundant, boring terpenes as entry points to a whole ecosystem of new chemistry, with microbes as your synthetic partners. By demonstrating that Streptomyces can carve abietic acid into multiple, structurally distinct derivatives, McCadden and co-authors give both chemists and biotechnologists a concrete example of how to fuse natural product biocatalysis with sustainable feedstocks. The study does not yet answer all the engineering questions about titers, productivities, or plant-scale economics, but it lays the molecular groundwork that those efforts depend on. For researchers, founders, and investors tuned into the bio-based chemicals wave, this is a call to look again at resin acids and ask, what else can we make from this cheap, sticky carbon if we let Streptomyces do the hard stereochemistry? For a follow-up episode, would you be more interested in exploring how to scale these Streptomyces biotransformations for hydrophobic terpenes, or in mapping the medicinal chemistry and screening opportunities around abietic acid-derived scaffolds?
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Ernala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.